Thank you, Mark, for a very kind introduction, and what a joy it is for me to be back here at Rackham, surrounded by people, some of whom I know, but many of whom I knew are doing great things here in biomedical research. I've already encountered some of my former colleagues on the faculty, some of my former postdocs who are now on the faculty, and a variety of other folks that are near and dear to me, because I feel once you've gone blue, you're going to stay blue. And I, too, want some of those ice cubes, because that would be really nice to show to those NIH folks who still aren't quite clear what the significance is of Michigan and blueness. But I know about that. And again, thrilled to be back here. And I'm a big fan of what you're doing with this MCubed program, which I've watched from a distance and had a chance to learn a bit more about in the process of coming here today. And you're probably aware that the rest of the world has also been looking at this program as one of particular innovative creativity in terms of how to start collaborations quickly without long delays, and how to catalyze those kinds of collaborations that might otherwise have too much of an activation energy. $60,000 is a great way to get over an activation energy problem. In fact, I've often said that one of the challenges we all have in medical research is that it often feels like herding cats. For me as NIH director, when there's a new opportunity to push science forward and people aren't quite lining up, somebody will generally say, well, what do you expect? It's like herding cats. And my response to that is, you're right, but you know what? I've got a big bag of cat food. That's sort of $34 billion worth, actually, and amazing what that can do. And you know, you have maybe not quite that big a bag, but this whole MCubed program can get the cats moving, and great things start to happen as a result. And you're going to hear about many of those in the course of the afternoon, these TED-style talks, and then all those posters. What a wonderful outpouring from MCubed 2.0, and I'm delighted to hear that 3.0 is going to get going next year, and I hope everybody in here is really already thinking about ways that you can tap into that opportunity. So what to talk to you about? Of course, I want to talk about collaborative science. It seems like the thing to do at a time like this for an audience like this. And I don't think I have to go over in great detail why NIH is an important player in this. Most of you are fully aware of that. Keep in mind that we have this dual mission of both basic science, which is led, by the way, to, at last count, 153 Nobel Prizes for NIH-funded investigators, including 4 just this past fall. Almost all of those for basic science. But we also have the interest and the obligation to move that into applications, and that's the other part of the mission on which we spend a little less than half of the budget, namely the applications. And as you all know, the money doesn't all get spent in Bethesda, although we do have a vibrant intramural program there that my lab is happy to be part of. Most of it goes out across the country to scientists. Here you can see the depth of color of the states tells you the relative proportion of funds that go to those states, and of course one of them is Michigan. And you have done pretty well here. In current fiscal year, 1,580 awards, $687 million to the state, and of that, U of M gets the lion's share. So you guys are doing a pretty good job here of convincing peer review that some of the cat food ought to come to Michigan, and it has been coming quite nicely, and we're thrilled about that. But what about MCubed? I'm fond of this quote from Woodrow Wilson: I not only use all the brains I have, but all I can borrow. From my own perspective, all the science that I've had the privilege of doing over these last 33 years as a principal investigator of a research lab, this has been true in almost every instance. Find out who's out there who has a complementary approach to something you're trying to tackle. Tackle a hard problem, as you just heard from President Schlissel, but don't imagine you can necessarily do it all by yourself. Find other partners that can work with you. And then it's both satisfying because you learn new stuff, it's fun because you're interacting with people who know things you don't know, and you make progress at a rate that you wouldn't otherwise. And it does seem to me MCubed has turned out to be an impressive example of how that can work. So just looking at the statistics so far, and again, this is still an experiment in progress, The numbers I have is that the projects funded by MCubed since 2012 have led to an estimated $92 million in external funding that has flowed to projects that started as MCubed. And I'm sure the leaders of the institution are looking at this closely, because this is what you call your return on investment, and that's pretty darn impressive. I just give you 3 examples there of projects that are supported by NIH that began as MCubed, and I could have given others, but those are 3 that just got picked out of the mix. So I'm sure there is hope that many of the projects you're working on today will find their way down that same path. Not all will, of course. When you tackle something like the sorts of projects that are being supported by MCubed, and of course many of them are well outside of biomedical research, some of them will lead on to other sources of support. Others will be self-contained and will do something on their own that doesn't necessarily go to that next step. But it's great when that does happen, and I'm sure there will be examples of that represented on the course of this afternoon. So what to tell you about where we are with NIH and how this attitude about collaboration is playing out, I figured I would divide things up in a brief presentation about 3 different themes. And you might wonder, what are the fireworks over there in the corner? Those are actually microtubules that are spontaneously forming in an extracellular system that both is nice biology, but it also makes a really nice visual. So first we're going to talk about uncovering life's foundations. And this is the basic science part of what I want to say. And of course, this is an enormously broad landscape, and I'm only going to pick a couple of examples to show where we are. And I'm going to start with neuroscience, because the current effort, in a very collaborative way, to engage the talents of people from multiple different disciplines, from engineering, from robotics, from computational science, from biology, from medicine, all focused on trying to understand how those 86 billion neurons between your ears do what they do, is one of the most ambitious and exciting enterprises of the modern biomedical research era. And we're just bold enough to think we might be able to start to figure that out. Not one neuron at a time, but actually how the circuits that involve maybe hundreds of thousands or millions of neurons in real time are able to do remarkably complex things, like processing visual information, like initiating some sort of voluntary motor movement, like laying down a memory and then retrieving it. I believe it's fairly likely that in another 8 or 9 years, we'll have some pretty interesting models of how those things work. And that will be transformative for our understanding both of normal function, but also as a foundation upon which we ought to be able to build more understanding of conditions like autism and schizophrenia and Alzheimer's disease. So Brain Initiative, big, complicated sort of human genome project kind of effort, although I would say a lot more complicated than the genome project was, but bringing all these disciplines together. And if you go to one of the brain meetings, it's like amazing, the conversations that are going on between people who had maybe a different language, but they're beginning to understand each other. So the focus, again, on circuits and networks, understanding how things work in real time, how these complex functions of our own brains and that of model organisms can be understood. And again, this isn't all getting done in one place. It's being done all over the place in the country, and I have to point out some of it right here at Michigan. If you look at our most recent awards, one of them right here, just announced in the past week, going to an investigator here at the University of Michigan, now part of this team of hundreds of investigators that are pursuing this together. And out of this, all kinds of interesting publications pouring out in terms of advances that we're beginning to understand. At this time, over 260 such publications that are coming forward, and we're really just getting started. This initiative is only in its 3rd year. So a good example of a collaborative enterprise. Let me give you another one just sort of getting started. Something called MotorPack, the Molecular Transducers of Physical Activity Consortium, which aims to try to understand how it is that exercise provides the benefit to human health that we know empirically it does, but we don't really know how that works. And not just about things like muscle strength and bone strength, but also about prevention of other illnesses, even about maybe improvements in brain function. There must be some molecular transducers of those benefits, but we don't know what they are. So this is one of those projects that brings together people from various disciplines to support their effort. 5 interrelated components, as you can see there. And again, guess which university is playing a significant role in this? That would be the University of Michigan, in this case, Charles Brandt and Jun Lee, who are part of the chemical analysis site, and we're counting on interesting things to come out of this, and I'm sure they will. Well, those are some of the very simple examples that I could pick of collaborative efforts in basic science. But now let's move to the second phase of this, which is translating discovery into health. And now you might wonder, what is that video? So this is a fascinating and beautiful example of how, with various dyes and microscopy, you can see what's really happening in a very interesting situation. The red cells are cancer cells. The cells stained in green are cytotoxic T lymphocytes that are actually going after those cancer cells. And every time the cancer cell flashes red, the lymphocyte just perforated its membrane and it's on the way out. So this is cancer immunotherapy being carried out in a carefully observed circumstance in a culture dish. But this is also an area of enormous current excitement, but one which is only going to make progress if we bring together the experts in cellular immunology, the people who understand the cancer cell at the most basic level, and the physicians who are poised to try to incorporate these new ideas into treatment. And a lot is happening here. The cancer moonshot, pioneered over the last couple of years with lots of support from former Vice President Joe Biden, and now at NIH, the source of an additional $1.5 billion of research money over the next 10 years, is focused almost explicitly on collaboration and how to bring these capabilities together, and also how to inspire people to be open about data access, a critical issue for all of us. The time has passed for investigators who have generated data that would be useful to lots of people to hang on to it for long periods of time. That's not the way that we make progress, and the Cancer Moonshot is certainly pushing hard about this issue of data access, cooperation, collaboration. And as an example of not only bringing academics together in terms of how to achieve that kind of collaboration, we very recently have announced just 2 weeks ago an unprecedented partnership between academic investigators and 11 pharmaceutical companies that are also working in oncology to work together on cancer immunotherapy, and particularly to identify biomarkers that might predict whether a particular immunotherapy approach is going to work for a particular patient with a particular type of cancer, because there's a big unknown about that part of it. This is pretty unprecedented. All the data will be made accessible to everybody. The companies don't get to hang on to it for their own private purposes, but they're willing to come to the table with their expertise and their dollars, because we all agree this is an opportunity to move the whole field forward more quickly. That's a kind of approach that I don't think would have flown 10 years ago, but it's flying nicely now because of the scientific potential and the sense that we can all get there faster if we work together in these sorts of pre-competitive spaces. There was a lot of buzz about the announcement of this, which is called PACT, the Partnership for Accelerating Cancer Therapies. And watch this space, because interesting things are going to happen. Let's pick another area of the application of science to human health. And now, if you'll forgive me, I'll slip into one where my own research lab has been deeply engaged in an area now for some 24 years, and that's diabetes. The adult onset or type 2 version of diabetes, which, as you can see from this graph, is showing dramatic increase across the United States and across the world. Much of this associated with an increase in obesity, which is a major predisposing component of how people get affected by this. But we've known that diabetes also has hereditary contributions. It tends to run in families, but not in easily understood ways. And so 24 years ago, right here at University of Michigan, as I was walking out the door to head off to NIH, Mike Benke, who's here in the audience from the School of Public Health, And I decided it would be appropriate to take some of those tools of genomics that looked like they were beginning to show the ability to find hereditary factors in well-behaved diseases like cystic fibrosis and Huntington's disease, and see whether it could actually be possible to unravel heredity in a very ill-behaved condition like diabetes, which we knew was far from something that Mendel would have approved of. And out of that came this collaboration called FUSION, the Finland-United States investigation of non-insulin-dependent diabetes mellitus. We don't call it that anymore. You can tell the project's been around a long time because its very acronym is outdated by the fact that we don't talk about NIDDM, we talk about type 2 diabetes, but the acronym has stuck. And again, one of the best ways to tackle a hard problem is to work with your friends. And Mike has been a wonderful friend throughout this, and we recruited some others. Jaakko Tumaleto, Markku Laakso from Finland, wonderfully capable clinicians who have provided us with awesome opportunities to do clinical research on individuals with and without diabetes in Finland, which is very well organized for this kind of research. Richard Bergman, physiologist of diabetes of known repute in Los Angeles, and my former postdoc, Karen Mulkey, now professor at the University of North Carolina, and others that have been added along the way, including Steve Parker, now assistant professor at the University of Michigan, Michael Stitzel, now assistant professor at Jack's Genomics, all of whom have come through this process as trainees and are now emerging themselves as fully empowered independent investigators, and a wonderful way to start a team. So what have we learned? Well, we've learned this is a hard problem. We are in fact in a position now of being able to have identified approximately 100 places in the human genome where there are variations that predispose people to type 2 diabetes. But somewhat to our surprise, and maybe you might even say dismay, most of those are not in the coding region of proteins. They are in the non-coding areas, in between genes, or located in introns, and therefore much harder to understand in terms of what the functional basis would be. And that is to say, if you want to understand hereditary factors in type 2 diabetes, you have to go beyond the protein coding knowledge of the genome to what you might call the epigenome, the other part of the genome that's involved in gene regulation. So to try to tackle that, and again, only possible because of a large collaboration over a long period of time, we are trying to understand in those tissues most relevant to diabetes, the pancreas, the liver, adipose tissue, muscle tissue, what is happening as a consequence of those risk variants in the genome, and how does that predispose somebody to the disease? And you can see down below a long list of the kinds of technologies, most of which have only been invented in the last few years, which can be applied to all those tissues and which together can teach you some very interesting things about diabetes, but actually in a basic science way about how the genome actually works, and how that other 98% of the genome that doesn't code for protein is actually responsible for a vast array of interesting biology and potential medical consequences. So some of the lessons, many of those non-coding risk variants from genome-wide association studies actually turn out to lie in long stretches of open chromatin, which we have called stretch enhancers. And that was a new discovery just a few years ago. And that these tend to be tissue-specific, these areas of open chromatin, and they tend to lie somewhere near genes that are also tissue-specific. But it is not easy to figure out exactly which gene is being modified by which enhancer. It isn't necessarily the one that's closest. If you just think of DNA as a one-dimensional structure, we have now been very much catapulted into the era of thinking as the three-dimensional nucleus, or maybe even the four-dimensional nucleus because of time as that fourth dimension. And that is fascinating and has inspired a whole host of new kinds of technologies to be developed. It turns out that once you begin to make sense of this, the risk factors are mediated then by specific transcription factors, proteins that bind to a specific DNA recognition site, and we can even in some instances say exactly how that risk spelling in the genome has altered that binding so that the gene that's modified is either up-regulated or down-regulated, and that in turn plays out in terms of explaining the risk. And you can imagine how that, because it tells you about molecular pathways you didn't know about, can lead to new ideas about therapeutics. So again, another example of how collaborative efforts have led to insights that never could have happened from us working by ourselves, and it has been a joy to be part of this kind of a multi-institutional, international collaboration that has sustained over all these years and produced all of these opportunities for insight. Well, the third theme is sort of a bringing together of a lot of things that I'm sure are on your mind and mine too, about how do we move this whole exciting process forward together. Because I think there's nobody who's involved in biomedical research right now who would say that this is a backwater time. This is like front burner excitement in so many ways, in so many parts of what we do. But how do we make sure we move that forward? Again, one tries to think of what's missing here in terms of new insights that our current approach might not derive. And for me, as the person who has the privilege of looking over the landscape and trying to understand what kinds of projects are we not doing that we should be, a big question is this whole idea about how do people stay healthy? Let's just not study disease, let's study health as well. And along with that comes the question about individuality. How is it that the individual circumstance may have information in it that you won't see if you lump everybody together? And this brings us to the whole concept of precision medicine, the idea that if you want to have the most effective ways of keeping people healthy or treating illness if it happens, it ought to take account of the individual. That one size fits all is not what you would do if you went to the shoe store or to get a new set of eyeglasses, and maybe it shouldn't be what you do when you go to the doctor. And yet oftentimes that's what we're stuck with. So I began dreaming, 20 years ago, about the idea of having a very large study of individuals, collecting every kind of data you could imagine and following them over time to begin to get that kind of data about individuality. And that led to a publication now in 2004, if you see the date there, where I tried to make the case for a prospective cohort study of genes and environment. Notice, environment. This is not just about genetics. This is about the holistic view of how environmental exposures, genetic factors, health behaviors, diet, lifestyle, all those things come to bear on whether an individual experiences good health or not. And the only way to really get those answers in a comprehensive way is to study a very large number of people. I proposed at that point that we might study 500,000. I don't think that was very well received, actually. That particular proposal landed with a thud. And it probably should have, because if you look at the comparison of where we were in 2004 and where we are now, it was utterly impractical to imagine doing something at that scale 13 years ago. But it's not impractical now. The cost of sequencing a human genome, which was about $22 million in 2004, is now, by most estimates, about $800. The time has greatly been reduced to do that. The use of smartphones as a means of collecting data in real time on people taking part in cohort studies has provided a brand new opportunity here for data collection. Electronic health records, even though they're clunky, have become available for research and have opened up all sorts of new windows that paper records would not have allowed, and computing power has advanced. So we have things that are happening. Patient partnerships are developing. People are more interested than they were in taking part in such research studies, interested in not just being human subjects, but being participants. Electronic health records, technologies like the kinds of things that many of you are walking around with. I have a Fitbit and an Apple Watch, and they're keeping track of me right now. Genomics, of course, not just DNA sequencing, but also looking at gene expression and other kinds of omics, and the opportunity to think of this as a really interesting data science challenge as well, recognizing that if this is going to be useful, these kinds of data sets need to be accessible to lots and lots of people who have great ideas about what to do with them while still protecting patient privacy and confidentiality. Big challenges, big opportunities. Well, it's happening now. The program called All of Us, which is currently in beta testing, has in fact been mounted with the strong support of a lot of very smart people as advisors. And I've recruited to lead it Eric Dishman from Intel, who ran their health division and knows a lot about large-scale projects and data management. The goal here is to really accelerate health research and medical breakthroughs across multiple different areas of medicine, to nurture relationships with participants so they really are the partners, to build a data set that's easy to access, and to really create this new kind of ecosystem for doing research. It is intended to be a very large-scale effort, enrolling 1 million or more participants who are consented and engaged to provide data on an ongoing longitudinal basis, including electronic health records, blood samples, which can be used for whole genome sequencing when that becomes affordable, wearing all kinds of wearable sensors to keep track of what's happening, and answering lots of questions about their own health and medical experiences. And the goal here, because we're very interested in using this as an opportunity to understand health disparities, is that more than half of the participants will be from traditionally underrepresented groups. And that includes both ethnicity, but also socioeconomic status, and having ample representation of rural individuals who often are left out of these kinds of studies. That's going to be challenging to make this all happen, but we are well on the way in the beta test phase to see how this is going to work. If you want to be part of this, you could either sign up as a direct volunteer, which is what you see on the left, or we've also signed up 10 large-scale health provider organizations who are reaching out to their participants to invite them to take part. And you can see at the bottom the kinds of things that we will collect on the individuals. And we already have set up, through a series of grant and contract applications, a national network of inaugural partners. There are about 100 organizations altogether already involved in this, which means it's a lot of moving parts, but it actually is coming together quite nicely. And as of May, we started the beta phase of testing. We have, as of last week, enrolled about 6,500 people as a test for each of these particular organizations to see if all the pieces are working together, doing some tweaking of that to be sure things are efficient, to be sure the database is highly secure. So far, looking quite encouraging, and we look to ramp up over the course of the next few months to all of those places, more than 100 locations across the US, testing all the methods. And then with the national launch coming up in the spring, at which point we will be blanketing the airwaves with opportunities to join up so that people hear about it. But if you want to know what's happening about this, www.joinallofus.org will allow you to sign up to get regular updates about where the project is and notifications if you decide you want to join coming in the spring, how to do that. This will be a platform for asking all kinds of interesting questions in medicine, because while we'll have basic data on these 1 million people, if you want to do an additional study on, say, diabetes or Alzheimer's disease or asthma, you will have a long list of people already with a lot of data collected who are pre-consented for recontact to take part in a disease-specific study, which will speed up the process of enrollment and greatly, I think, make the whole process more efficient. And so both academics and companies are extremely interested, I think, in seeing this emerge as a way to catalyze a whole lot of additional research. Now, by talking about all this, I'm coming to Michigan, and I have to admit that you guys have thought about this before all of us came along, and so I have to do a shout out to the Michigan Genomics Initiative, which has already enrolled 42,000 plus participants in a remarkable team, some of the members of whom you see listed here and some of whom are in the room. And we are counting on that particular initiative as a partner that we can learn from, and certainly we've had great assistance from people like Gonzalo and Sachin as advisors about how to set up the national program. If we want to talk about moving forward together, just the last couple of things, we also really have to think about the most important resource for the future of biomedical research, and that's the people that will have the insights, that will do the work, people like all of you in this room, people who will join MCubed projects and will come up with creative approaches to answering questions that maybe we didn't even think we could dare pose a few years ago, but maybe now we could. And we at NIH are very interested in supporting that kind of thing with high-risk, high-reward programs like Pioneers and New Innovators and Transformative Research Awards. If you don't know about those are programs where you don't have to have a lot of preliminary data, but you need to have a big idea. And if you bring that forward and convince peer reviewers that your big idea is worth pursuing, you get a substantial amount of funds over the course of 5 years to go after it. And some of our most exciting insights have come out of those programs funded by the Common Fund. We're also intensely interested in expanding the diversity of our workforce because we know that teams are more productive when they are more diverse. That's well established scientifically, and frankly, we are still short of the kind of diversity that would be healthy for our workforce across this nation, and NIH has some new ideas about how to support that, how to make our workforce seem more welcoming to people who have traditionally not been well represented. We're also concerned about the slide in physician scientists that are doing meaningful efforts in research, by which I mean spending a significant fraction of their time, maybe a half or more in research. And we have new programs to try to encourage physicians who are interested in research to have pathways forward that make that possible without being sort of seduced away by clinical medicine, which is also a great thing to do, but we need physician scientists, especially now with all the opportunities. And we're doing something right now that aims to try to provide a substantial increase in funds for those individuals who are just starting their career or who are in the place of mid-career investigators who need to keep their labs going or perhaps even expand into new territories, something called the Next Generation Research Initiative. If you haven't heard about that, it's been written about a bit in the pages of Science and Nature, and you can read about it in the websites for NIH. So particularly if you're just starting out as an assistant professor, this is a good time because we are really seeking ways to be sure that that support, which has admittedly been struggling a bit over the last 10 or 12 years, is coming back to where it should be. I'm really pleased to see that MCubed has taken this idea of supporting trainees in a very serious way, with 50% of the funding going to undergrads, grad students, and postdocs. I don't know who made that decision, but my hat is off, because that's the future, and that's where a lot of the exciting science ends up getting done on teams, because they include folks of this sort. Final thing before I conclude is just to remind all of us that in order for these exciting times to go forward, for research, collaborative research in particular, to succeed, we need a steady flow of support. And this has not been an easy 15 or so years for that. If you look at this graph, this shows you what the funding has been from NIH since about 1990. The white curve there shows you what the actual purchasing power has been for NIH if you adjust for inflation. The dotted white line is the Recovery Act, which was a 2-year infusion of money, but that's now quite a while ago. The dotted yellow line is what would have happened if we'd stayed on the same trajectory that we were on starting back in about 1970 and just kept that going. But that isn't what happened. In 1998, we had this wonderful 5-year experience of a doubling of NIH, and you can see how the white curve went up fairly steeply. And by 2003, success rates for grantees were at remarkably good places. Maybe about 30% of grants were getting funded. But you can see what happened after that. A gradual slide as inflation began to eat away at stagnant budgets. Really bad things like sequestration in 2013, further damaging the support for medical research. And by the time we got to 2015, the success rate for a grant applicant had dropped down to about 16%, about half of what it had been. And all of you who are involved in research at that point will remember that this was a pretty tough time. The good news is, and I hope you can see that from this graph, is that we hit the bottom and then things have started to come back in the positive direction. And much credit to the leadership of the United States Congress in both houses and both parties who have, in fiscal year 16 and 17, found a way to give an additional $2 billion to NIH for each of those years, and who seem poised, we hope, by December of this year, to do that again for fiscal year 18. That would mean, if it's successful, something like a 20% increase in support for biomedical research in a 3-year period. And that should give all of you encouragement that perhaps a difficult time that we went through from 2003 to 2015 has now kind of gone by, and we're in a much more encouraging opportunity here for something that can encourage stable growth and an opportunity for people to take risks. So if you haven't heard about that, because sometimes the memory of hard times lingers even longer than the hard times, My grandparents grew up in the Depression. They still were living in the Depression in the 1960s. Sometimes we in academia have that same problem. We're not in the Depression, folks. This is actually, I think, a pretty exciting moment to see how the enterprise is moving forward, and therefore to be bold with ideas. Many of them coming out of MCubed, I suspect. Bring those bold ideas to NIH. We're hungry for those. We are always trying to make our peer review system better so that it embraces innovation and novelty and doesn't ding you if your budget seemed a little out of whack or if your buffer had the wrong concentration of sodium chloride. We're really interested in those ideas that I know must be bubbling up in the brains of the people here in this room. And my hope would be that you would see this as a great opportunity to move our whole field forward. I wear this button on my lapel, which says hope at NIH, because we often think of NIH as the National Institutes of Hope, because people think of us as creating that hope, both in terms of understanding mysteries about life, but also finding answers for medical problems. But Peter Levi wrote this wonderful quote, which I'm fond of, that hope is in every sphere of life a privilege, not just something you throw out there. It's a privilege, and it attaches to action. No action, no hope. If we want to see advances in life science, if we want to see advances in science in general, if we want to see advances in our society, it requires that kind of action. I know MCubed is about action. NIH is all about action, so we're happy to be your partners. Thank you all very much.